Okay. Hello, everyone. My name is Peter Bannattini. I'm at the National Institutes of Mental Health. I'm also the editor-in-chief of NeuroImage, and I'd like to welcome you to this webinar hosted by the journal NeuroImage entitled Multiscale Optical Imaging and Microscopy of Neurovascular Coupling in the Living Brain. I'd also like to thank our sponsors at Andor for making this event possible. So before we begin, let me remind the audience that we have a questions and answer session following both of the presentations, so at the end of both of the presentations. So please submit your questions at any time during the presentations using the Ask a Question button on the right-hand corner of your screen. I'd encourage you to input questions as and when you think of them, and they will be addressed in the QA session at the end. The more questions asked, the better the session will be. So it's my pleasure to introduce and welcome today's first speaker, Dr. Elizabeth Hillman. So Dr. Hillman is an associate professor of biomedical engineering at Columbia University. She received her undergraduate and PhD training at physics at University College London and was a postdoctoral fellow at the Massachusetts General Hospital Martinos Center before joining Columbia in 2006. Dr. Hillman's laboratory has a dual focus on the study of neurovascular coupling and the development of novel optical imaging tools to study the brain. So without further hesitation, I'd like to hand over to Dr. Hillman to begin her presentation.
Thank you. Okay, so welcome. I'm going to talk to you, as Dr. Bandattini said, about both the development of optical tools for looking at neurovascular coupling and also neurovascular coupling. So I will start with a brief introduction of generally sort of what is neurovascular coupling. And so what we're thinking about is blood flow in the brain. And what a lot of people don't necessarily realize is that as neurons fire in the brain, you tend to get an increase in blood flow to that region. And the brain is constantly changing its blood flow to the local region of the brain according to when neurons fire. Now, this change in blood flow is actually the basis of functional MRI. So this is the signal that's actually detected when you do an fMRI bold scan. The signal that fMRI detects is actually the deoxyhemoglobin signal changing here, because deoxyhemoglobin is paramagnetic. And so when you see these sort of red blobs in these MRI images, what it's telling you is that the a level of deoxyhemoglobin actually decreased in that region of the brain. Now, this is a little bit curious if you think about it, because most people would assume that the blood oxygen level dependent signal would represent a consumption of oxygen by active neurons. But what it actually represents is an increase in blood flow to that region, which causes an over-oxygenation of the region. It's also kind of a curious response, because that increase in blood flow happens much more slowly than neurons firing. So here we have a plot of calcium activity that's showing that the neurons are firing in response to this stimulus much more quickly than that blood flow increase is happening. So it's sort of a mysterious process that's happening in the brain. But the interesting part is that we know that it must be essential for normal brain function, but we don't have a lot of knowledge at this point about what mechanisms are actually driving that blood flow increase. And so here's a schematic of all the different kinds of cells that we have in the brain, and almost all of them have been at some point implicated as being involved in signaling between when neurons are firing and causing blood vessels local to those regions to increase their blood flow. So we're really interested in this because it's obviously an intrinsic, very important component of normal brain activity, and we currently have very little understanding of why it looks the way it does, and that also reflects on being able to properly interpret the signals that we're able to measure using functional MRI, both in the normal brain and in the diseased brain. So if we want to study this, we need to simultaneously be able to look at neural activity and blood flow, all in the intact brain, which makes it a very good imaging challenge, which pairs this research very well with the development of new techniques to be able to actually capture the dynamics of this at lots of different length scales. So what actually is happening in the brain during this response? So what we can do is actually take an animal and expose its cortex. So this is actually just a camera photograph of the surface of the brain in a rat where we've illuminated using green light. Now, over here on the right, we have the absorption spectrum of oxy and deoxyhemoglobin. And what that tells us is, and what a lot of people know, is that red blood, very red blood, is oxygenated blood, and deoxygenated blood is more of a brown color. And so if we actually measure this image using multiple different colors of light, we can actually create an image that corresponds to the oxygenation level of the blood in the picture. In this case, we've just artificially colored the arterioles red because they have a higher oxygenation level than the veins that are blue. And we can turn this kind of data into a nice movie that will actually show us, when we stimulate this animal's paw, what happens to the blood flow in the brain at this level. So this is like the fMRI bold response, only zoomed in and at very, very high acquisition speed. So what we're going to see is here, this is the total hemoglobin, oxyhemoglobin, and deoxyhemoglobin that we have calculated based on the different colors of the blood. And what you can see is this very sort of interesting, complex relationship in the way that the brain is selecting to dilate the artery. So you just look again, if you look over here, you see kind of blushing in the capillary bed, followed by this rapid dilation, sort of spreading dilation of the arterioles on the surface. And over here, you can see that the majority of the deoxyhemoglobin signal is coming as a decrease in the deoxyhemoglobin in the veins, which corresponds to a washout of that over-oxygenated blood supplying the region. Just to sort of provide a little bit more information about how we got this data, we developed this system to permit us to get very, very high-speed multispectral imaging. So what we wanted to do was sample the image using 3 different wavelengths of light, so a blue wavelength, a green light wavelength, and a red wavelength. And traditional ways of doing this would use a filter wheel spinning in front of a white light source, and that would trigger a camera to collect an image. But cameras don't really like to be triggered. Cameras like to free run. And so we're able to use either the DALSA 1M60 here or the Andor Ixon 897, and that can output an actual pulse that represents when it is free running and acquiring an actual camera frame. We can use that pulse with a little bit of hobby electronics to drive these different LEDs to flash only when it's grabbing a frame, and we can sequence them and cycle them however we want. And that gives us the ability to acquire these kinds of images at very, very high speed. So we can go up to about 200 frames a second, which then for 3 wavelengths would come down to about sort of 65 frames per second, and we get all 3 wavelengths, which we can then convert into that hemoglobin spectrum. So this is a very helpful system. We've actually made this into a software package, which is open source and is available to download. You can set it up with a simple FireWire camera. If anyone's interested, please check out my website. Now, this kind of imaging is a little bit confusing to understand exactly where the signal's coming from. I showed you kind of a 2-dimensional image. You're illuminating the surface of the brain, but the light is actually scattering down into the tissue and coming back out again. So you're getting signal from down to layers probably about 500 microns into the tissue. So it's superficially weighted. You're getting a lot of signal from the surface, and when we use these visible wavelengths, we see those surface vessels in very dark contrast. But when you look at an image from the top here, when you see those vessels, you know you're looking at the vessels, but these kind of fuzzy signals that you see underneath are coming from the capillary beds underneath, and there's a little bit of scattering there that makes the resolution there worse. Okay, so we can analyze this kind of data. These data sets can run up to over 50 gigabytes a day in my lab, and you can analyze these data in all different ways. So this is just a still showing the changes that you get and showing, again, those dilations of those arteries on the surface. But we've done a lot of work here, actually published in Neuroimage, looking at the onset dynamics and the decay dynamics of this response to really try to understand exactly where does it occur, why does it occur, how does it occur. We wanted to benchmark this response in order to figure out what kind of mechanisms could explain the behavior that we were seeing. In this case, we're stimulating the hind paw or the forepaw in this same animal, and we're able to see that there's a directionality to the way that this vessel dilates. And when we stimulate at one end of that vessel or the other end of that vessel, the direction of that propagation actually completely reverses, which tells us that this has got nothing to do with the direction of flow in the blood vessel. So these properties allowed us to really, like I said, benchmark. We see this propagation, and we see that it's traveling at very high speeds here, around 4 millimeters per second, in a direction that's independent of flow. And so we really wanted to try and understand what could be causing this. Now, one of the leading hypotheses related to how neurovascular coupling works is that astrocytes are involved. And so this is a schematic of a diving arteriole coming down into the parenchyma of the brain. And we already know that there's lots of different kinds of cells that connect onto here. And astrocytes really are known to be glomming all over the diving vessels and the capillaries of the cortex. In a seminal paper in 2006, Microscopy's group actually used this kind of imaging, used two-photon microscopy to image astrocytic end feet around diving arterioles here, and did calcium uncaging, and it was able to show that you could see increases in intracellular calcium in an astrocyte when you did uncaging, and a corresponding dilation in the blood vessel. And this led to a lot of people really concluding that astrocytes must play a role in turning on this blood flow change. But in our data, we see these sort of very complicated sequences of evolution of this response. And like I showed you at the beginning, we see that sort of the response is starting here at the capillary level and seems to be spreading up the arterioles. And from histology, we know that there are no astrocyte end feet actually connecting with arteries on the surface of the brain. So we were looking for a more sort of holistic solution here that would explain all of the properties, all of the dynamics and the speed of this response, that would be consistent with the morphology and the functional behavior of the different kinds of cells that we were suspecting as being involved in neurovascular coupling. So one way that we can do this is to use two-photon microscopy and also here calcium imaging. So what we wanted to do here was just simply get a better idea of what was going on neuronally and what was going on hemodynamically. We can do that with our system that I already described with a very simple modification to just add a long-pass filter in here. So what happens is when the blue light is on, you're exciting this calcium-sensitive fluorophore, and you're getting green light emission to your camera, and so you get this fluorescent image of the surface of the brain. Under the green light excitation, you get reflectance because that green light can go through the filter, and you get hemoglobin measurements. And so this is data from quite a long time ago where we actually did bulk loading of Oregon green into the cortex, and we can get recordings like this. So on the left here, you see the hemodynamic response. On the right, you see the calcium response. And you can see that as we're stimulating the animal's paw, we see this bang, 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 bang neural response, while we see that much slower hemodynamic response. We use this to study the role of astrocytes by basically taking an animal, looking at this response in this wide field view, and then taking that same animal into the two-photon, into our home-built two-photon microscope, and we label the astrocytes with sulforhodamine, which is pink, and the neurons are labeled also with the Oregon green, which also loads the astrocytes. And when we do a stimulus now, we can actually look at the dynamics of the Oregon green signal here in the neuropil and in the neurons, and we can also isolate out the signal in the different astrocytes. And what we found here is that mostly the astrocyte signals that we saw were very slow and very sluggish in relation to the neural responses that we were seeing. And in this same animal, we know that the hemodynamic response is starting all the way up here, which is much faster than these astrocyte responses that we were observing. Now, we did this study as a fairly small study. Nizar et al. Published this in J Neuroscience in 2013. But they did this very comprehensively throughout the cortex and found the same thing, that the astrocyte response seemed to be much slower than the neural response, which just gave us pause to think, well, is it possible that the astrocytes aren't the only thing that's involved in generating this hemodynamic response? We ourselves did another study using these techniques to just explore generally the astrocyte morphology within the brain, because, again, when people do immunohistochemistry, you tend to lose track of where you are in the brain, and people rarely look at the surface vasculature. So this is a two-photon microscopy image actually acquired in a live mouse, where we've labeled the white here is actually sulforhodamine in the astrocytes, and the red is actually Fitzy in the vasculature. And this is a movie showing that as a serial section going down into the brain. And this permitted us to actually really analyze exactly where were the astrocytes, what were they doing, how did they ensheathe the vessels, and all the different properties. We concluded here that astrocytes really do love blood vessels. They're everywhere. But we didn't find any connections to the PL arteries, as I said before. We found that they loved arteries, capillaries, and veins equally. So although they were around the diving arterioles, which people have interpreted as a sort of form follows function relationship, which means they must be involved in the dilation of arterioles, they were also wrapped around capillaries and venules, where we wouldn't expect necessarily the astrocytes to be playing a role in changing the diameter of the vessel. And so this still left us with the question, well, we know that the PL arteries dilate. We know that they do so very quickly, and we haven't right now got a mechanism that would explain how those vessels were dilating. And since they dilate very quickly, we thought they may explain what was going on with that early stage of the response. And so one thing we started thinking about was, what if the signal that was getting from the firing neurons down here at the capillary level up to the PL arterioles wasn't spreading within the cortex itself, but was spreading along the blood vessel? And the idea that it was spreading along the blood vessel was very attractive because it sort of gave the indication of some sort of electrical circuit that could very rapidly recruit the blood vessels needed to feed the region that was active. And so I sort of started searching the literature trying to find, what had been done to understand the different ways in which blood vessels might be able to propagate signals. And so this is, again, just some sequences from a movie of our data, and it shows this same thing, that the first thing that we really see is that these distant arterioles dilate, followed by a kind of blushing of the cortex. And then actually at the end, you see a residual blushing of the cortex with the arteries on the edges not dilating. So we found some work by Michael Kotlikov's group and Steven Seagal's group, and this is actually an animal that's been prepared. This is in cremaster muscle. This is not in the brain, but this is an animal that is a connexin 40 GCaMP2 mouse, which means that it's expressing a calcium-sensitive GFP in its endothelium. And what they show is if they puff acetylcholine down here onto this blood vessel, they observe two different kinds of propagated vasodilation, which is actually spreading along the endothelium of the vessel. Firstly, what you'll observe is as soon as they puff the acetylcholine, this vessel over here will dilate almost immediately. And then secondly, you'll see a calcium wave that will slowly chase that initial dilation. So let's have a look at that. Okay, so the stimulus is now, you see that instant dilation up here, and then you see that subsequent propagation of the calcium wave. I'll play that one more time. And so we've got these two components, this fast EDHF-type component that can spread very rapidly over a millimeter away within a few milliseconds, a few hundred milliseconds. And then you've got this slower calcium wave that is more, it's nitric oxide dependent and prostacyclin dependent, and it's more of a creeping wave with a slower spatial extent. And if you look up here, this is a data, as we pull data out from our data sets, what we observe is that that long-range signal that we see to the arterioles increases very quickly and then goes basically back to baseline quite quickly. But this slower component that's more local to the parenchyma seems to be sustaining for longer. And we think that those properties actually are very consistent with these two different properties of endothelial propagation in blood vessels. So we're very excited about whether or not the endothelium itself was contributing to the way that the neurovascular coupling response was being generated. So we need to figure out how to use optics again to do a good job of figuring out whether or not this was really happening. And so we used a technique called light dye, which is about 50 years old, but is far more specific than some of the genetic modifications that people are using today. So what you're able to do is if you put fitzy-dextran, so fluorescein conjugated to heavy dextran, into a blood vessel, it stays within the blood vessel. If you shine light onto that fitzy-dextran, it actually creates free radicals, which directly affect the endothelium, which is on the inner surface of the blood vessel. And if you do it for the right amount of time, you essentially damage the endothelium very selectively, and you leave everything else intact. Because we're able to do light-based treatment of this, we can pick pretty much any region of the vascular tree that we want to selectively damage the vascular endothelium in. And so in this case, we used what we called a light line, which was just a focused line of blue laser light to selectively interrupt endothelial signaling in a given vessel. So this is the response that we see when we stimulate the animal. We see these arteries all dilating here in response to a hind pause stimulation, and we chose this vessel and we tried to truncate the endothelium just at this point. So when we do that, we found exactly what we expected, which is that on this side, the signal no longer gets to the vessel and the vessel doesn't dilate. Now, if we put acetylcholine or sodium nitroprusside on here as a control, this side does dilate. So what it tells us is that the signal is trying to come up from here, and it gets to here, and it can't pass because we've selectively damaged the endothelium. And so we see this basically on one side, and the other side we see that it doesn't get past that point. If we actually shine the light over the entire region that's responding, we can essentially knock out all of that dilation of those PL arteries. And so that has a strong effect on the bold response, and actually seems to have the strongest effect on attenuating that early component of the hemodynamic response. So this completely convinced us that the vascular endothelium is acting as a conduit for conducting the hemodynamic response, and therefore that the endothelium is a really important component of this process. And so, I'm not going to show this for very long because this is a complicated diagram, but this has got us very interested now in really looking at the mechanisms of endothelial signaling and how that fits into the big picture of what we've been thinking before in terms of neurovascular coupling. So this is the astrocyte on the outside of the vessel, and we've identified a lot of these pathways that appear to be involved in neurovascular coupling. But what we note is a lot of those same pathways are actually involved in endothelial propagation and endothelial mediation of vasodilation. So it's actually the endothelium here on the inside of the vessel is able to tell the smooth muscle cells from within to dilate, whereas most people have been concentrating their attention on the outside of the blood vessel. So that propagation of vasodilation provides a new explanation for prior results that have implicated astrocytes, because we now no longer need the astrocytes around the diving vessels to be dilating, or to be showing calcium transients at the same time as dilation if the signal that's dilating the vessel is actually coming from elsewhere and traveling along the endothelium. This could provide us with new things to look for in terms of what might be triggering that bold response. Because, for example, endothelial cells have receptors on them for all kinds of interesting chemicals like acetylcholine, bradykinin, adenosine, ATP, ADP, UTP. So there's a lot of new routes by which signaling could directly get into the endothelium at the capillary level. There's two types of endothelial signaling that I described, the very fast one and the slow one. We have been doing modeling, and it seems that they can actually predict a lot of these nonlinearities of the bold response. And finally, a lot of these pathways, like the generation of prostaglandins and nitric oxide, are a lot of the things that are actually targeted by a lot of common drugs that we have in our bloodstream. And people have been concerned about the effect of drugs that can cross the blood-brain barrier, but in this case, drugs could directly be affecting the inner endothelial layer of the blood vessels, which could be having a direct effect on neurovascular coupling. And there are studies that have demonstrated alterations in the bold response to a lot of different kinds of drugs and also disease states, but it's often assumed that that's because the neurons are being affected, and then the neurons are just simply driving some representative blood flow response. What we hypothesize is that in some of these cases, the blood flow response itself could be the thing that is being altered by these different disease states or compounds. And in that case, if you have a blood flow response now which is not properly tuned to neural activity, this could be a new route for cognitive decline and cognitive impairments that you see associated with things like systemic cardiovascular disease, diabetes, and even Alzheimer's. So this is an area now that we're studying in great depth, and I think Mark is going to talk a lot more about really teasing apart a lot of these aspects of endothelial function in the brain. I'll segue a little bit here. I wanted to talk a little bit about our other approaches to imaging neural activity. So I showed you a little tiny bit of wide-field Oregon green imaging earlier on. In the last few years, we've been very fortunate for the development of GCaMPs, which are, like I said, calcium-sensitive green fluorescent proteins. So it's now possible to breed animals that are expressing selectively this calcium-sensitive fluorophore in any cell. And one of the most popular ones is this Phi1 GCaMP, or really just expressing it in different kinds of neurons. And what that means is that you can actually just have a mouse, breed a mouse, and open its brain, and its neurons will actually be flashing their fluorescence in any time that a neuron fires, because during neuronal firing there's an influx of calcium, and the fluorophore sees that entering the cell. So we thought, well, it would be really interesting to see if we can image this, and this would give us the opportunity to use our optical techniques both for imaging the hemodynamics and for imaging the neural activity, which is particularly important if we want to start looking at disease states and the effects of different drugs on the coupling between neural activity and hemodynamics. And so again, we just do this in our system by putting that long-pass filter in here. So we use two wavelengths here to get hemoglobin, oxyhemoglobin, and deoxyhemoglobin. Then we can use the blue to excite the fluorescence. So this is an example of a system that we've recently set up using an EMCCD camera. which allows us to do 512 by 512 at 30 frames a second. And we're able to image both calcium and the oxy and deoxyhemoglobin dynamics. And we've even added speckle flow imaging to this same system. And this works actually in awake-behaving mice. So we're able to train the mice to just sit on this platform and have their brains imaged repeatedly, which allows us to look longitudinally at what's going on, as well as to deliver all different kinds of stimuli. And we actually expose almost the entire superficial cortex of the brain. So we can see all kinds of different cortical regions and record all of them in parallel. And this kind of ability to record neural imaging, neuronal activity over this length scale is fairly unprecedented. So what I'm showing you here actually is just data collected using this system. On the right, you'll see the GCaMP responses, and what you're going to see is just spontaneous activity. So this is an awake animal basically just sitting there, not doing very much, and you're going to see neural activity, and then you're going to see the hemodynamic activity on the left actually chasing that. And this data is very relevant to resting state functional MRI, where we're really interested in whether or not the fluctuations you see in resting state BOLD are all completely correlated to underlying neural activity. So you see these neural waves kind of coming and moving around the cortex, and you see the hemodynamics trying to kind of keep up with them. And what we find is that certain spontaneous events appear to be followed very well by hemodynamic responses, but others don't evoke the same kinds of responses. So we're very excited about this system. We've just recently got this up and running. Okay, so finally, I just want to do a little bit on a different technique. So now we've realized that there's so much going on in this neural activity, this spontaneous activity. We want to zoom in again and look at this at a cellular level. But the work that everybody's been doing on two-photon microscopy, and we ourselves have worked very hard to try and get two-photon microscopy to go as fast as we possibly can. The problem with two-photon microscopy is that you're scanning a single spot around within a volume. And so imaging a volume like this is about as much as we're able to do at the moment at cellular level resolution using two-photon, because you simply don't have enough pulses in your laser, and you're not able to sample points quickly enough to sample every single volume in this, every single voxel in this volume individually in sequence. And so we really have been scratching our heads for the last 5 or 6 years trying to come up with a way of doing this. And one thing we recognized was that light sheet microscopy in this standard configuration achieves some very nice imaging performance by being able to illuminate a plane and then being able to capture an image of that plane all in one go. So you don't have to visit each point in turn. You can take one picture of the plane at one point in time. However, the imaging geometry of light sheet here has always been very awkward and clunky. You've got this sort of right-angled two different illumination planes. And so in this case, it's being used for imaging a clear, dead brain. So we wanted to think carefully about how we might be able to do this both at very high speeds and also in a geometry that would be compatible with in vivo brain imaging. And we came up with this geometry. The paper for this just actually came out in Nature Photonics on Monday. So what we were able to do is design this geometry where a light sheet actually enters the tissue at an angle from the objective, and the light is being detected from that plane through the same objective. So you can stick basically any sample under here. And the first thing that we're able to show is that we can have a light sheet coming through into the sample, and we can image the plane on an angle. Okay, so we don't have to image a plane that is normal to the edge, to the face of the objective. You can actually image any plane through an objective as long as you don't mind the image that you get being on an angle as well. So we figured that part out, and then we're able to also scan that light sheet. So in being able to actually sweep that across the sample, we're able to capture a three-dimensional image as well. And so by both being able to image that plane through the objective and actually move it around, we're able to do very fast three-dimensional imaging. So this is the overall system. And right now we use this funny-looking polygon mirror. We actually have a lot of other ways of doing this now. But essentially, when we move this polygon mirror, it sweeps that sheet through the sample. And by descanning the light coming from that sheet on the same mirror, it creates a stationary image just of the plane that's being illuminated. So although that plane's moving around in the sample, the image that we get corresponds to the piece that's being illuminated by that plane, but which is moving through the sample. And so it's much like confocal microscopy, only imaging a plane. And so this means we can image anything, and we can do very, very high-speed imaging because there's no translation here. This objective doesn't move up and down. The only thing that moves is this polygon, and it just goes back and forth at the volume rate. So if you want 10 volumes a second, you just go back and forth at 10 volumes per second. So the sample doesn't need to be mounted or rotated or translated, and the objective isn't bouncing up and down, so you don't have any disturbance to your small samples. And the camera here, we're using an Endothelial camera, which we can read out at 2,400 frames a second with 80 rows, which gives us 80 depths in our volumes. So does it work? So I'll show you very quickly just some examples. So this is a volume acquired in a mouse brain. The green is GCaMP being expressed in the dendrites of layer 5 neurons. The red is dye in the blood vessels, because, of course, we do neurovascular coupling, so we're interested in that. And you can really get volumes here that look much like two-photon microscopy. Our penetration depth is limited right now because we're using a 488 nanometer blue laser, which doesn't go very deep into tissue, but we've done down to at least 150 microns in the brain. And down to about 300 microns, you can see the diving vessels. But we get really nice optical sectioning. And this is an example of actually looking at neural activity. And you can see the temporal resolution that we see in our data allows us to actually do analysis of the dynamics of firing of individual spontaneous events throughout the three-dimensional dendrites of these spontaneously firing neurons. So we're really excited about this technique because it delivers something that's way beyond what's possible right now with two-photon techniques. Plus, it's a relatively inexpensive system. We've actually also been using it, a little bit of a sidebar, to image small organisms as well, because we can essentially fit an entire organism within the field of view of the system and image it in 3 dimensions as it freely moves along, which is kind of a dream of the Brain Initiative, to be able to capture everything that's going on in a given organism. And we have a lot of movies and stuff on this on YouTube right now. If you're interested, please take a look. And this is another one where we're actually co-expressing GCaMP and mCherry in the same larva. So what you can see is as its muscles are pulling back and forth as this larva is moving its head in and out, we're able to both see the structure of those muscles and also the GCaMP showing the calcium activity in those muscles as the animal is moving. And finally, we've been able to implement this most recently to image GCaMP in zebrafish heart. So this is a zebrafish embryo, and this is actually the beating heart of a zebrafish. We've co-labeled, we can do 2 colors with this, so we've co-labeled the heart muscle with both GCaMP and DS red, which lets us really look at any feature of this beating heart that we want to at basically cellular resolution. And with that, I will leave time for Mark. So just to summarize, so this new technique, which we've called Scape, brings the potential for imaging both structure and function in freely moving organisms. In the awake mammalian brain, imaging is on the order of 100 times faster than conventional laser scanning microscopy with no moving parts. The system is low cost, simple to use, no sample preparation, a priori information is needed, nor any image reconstruction in the data that I showed you. And we also think this is very exciting because the objective doesn't move up and down. We're able to combine this with photostimulation, such as optogenetics, and so we should be able to target single neurons, stimulate them, and image the three-dimensional response all in parallel. With that, I will point you to my lab website, which has a lot more information about this work, and I'll acknowledge the contributions of my students and of my collaborators and my funding. So thank you very much.
Okay. Thank you very much, Dr. Hillman. And, yeah, that was a really nice talk. There's several questions that have come about, but we'll wait until the end to address those. But so without further hesitation, I'd like to introduce today's second speaker, Dr. Mark Nelson. Dr. Nelson is a University Distinguished Professor and Chair of the Pharmacology Department at the University of Vermont. He was also Professor at the Institute of Cardiovascular Science at the Manchester University and a visiting professor in the Department of Pharmacology at the University of Oxford. He received his bachelor's in mathematics and biology with honors from Tufts University in 1976 and his PhD in neurosciences from Washington University in St. Louis, Missouri in 1980 under Professor Mordecai Blaustein. He went on to receive postdoctoral training with Professor Blaustein at the University of Maryland and with Professor Lauger at the Universitat Konstanz in West Germany. So without further hesitation, I'd like to hand over to Dr. Nelson to begin his presentation.
Hi, everyone. That was great, Elizabeth. I really loved it. In knowing what she was going to talk about, I wanted to take a little different perspective and zoom in on the vascular endothelium and how calcium signals control vascular endothelial function. Initially, most of the work I'll talk about has been done in small arteries from resistance arteries from mesenteric arteries, but I'll show you how we're extending that into the brain endothelium, including the arterial endothelium within the brain and capillary endothelium. This is Vermont. Also, for reference, much of the stuff I'll talk about are in these publications. As you heard about from Elizabeth, the brain is highly vascularized, and this shows an example from David Kleinfeld's lab. And I've been trying to make some estimates, but my guesstimate now that perhaps the endothelial cells, the focus of Elizabeth's work, outnumber neurons probably 10 to 1, and guesstimates of endothelial cells in the brain, they put them at maybe at 1 trillion cells in the brain. They're contained they line all the blood vessels, the arterioles, and they make up the capillaries of the brain. And this shows you a little bit of background. All endothelial cells line all the blood vessels. They constitute about 2% of the body mass. There are probably several trillion endothelial cells in your body. The endothelial cells function as a barrier, so it's a blood-brain barrier in the brain. They signal to the adjacent smooth muscle to cause vasodilation. Endothelial dysfunction contributes to virtually all vascular disease. It's now appreciated that vascular disease is a major component of neurological diseases. And one of the prime regulators, which you heard about in the last half hour, is intracellular calcium. And I'm going to show you techniques for looking at intracellular calcium at the level of the single molecules and give you criteria and the implications of that for both endothelial function in the periphery and in the brain. And it's just an example of all the things that go wrong with the endothelium throughout your whole body. And as I also give in talks, you can just sort of tick through which ones are your experience at the moment, so it's important to keep your endothelium healthy. This shows a little blow-up of the architecture of the endothelium. The endothelium sends processes through the internal elastic lamina, and it contacts the adjacent smooth muscle, almost like a nerve synapse. And many of them have gap junctions, so the endothelial cells can communicate electrically to one another, so they're almost like an electrical wire through the brain, and they communicate to the adjacent smooth muscle and the arterioles electrically, as well as releasing factors. And I'm going to focus on this projection to give you examples of what's going on in the endothelium and how this is controlling vascular function. And these are called myoendothelial projections, and they have a variety of transport proteins that show them the EM blow up. They have gap junctions. And our previous work also showed that there's endoplasmic reticulum in the projections and a variety of ion channels, particularly the intermediate conductance calcium-sensitive potassium channel and TRPV4 channels, which I'll talk about, are localized and concentrated in these myoendothelial projections. And so we're really zooming into the microscopic level as opposed to Elizabeth's presentation. So most of the work I'll talk to you today will be on mouse mesenteric arteries, although we're doing the same things, and I'll show you some examples, in brain parenchymal arterioles as well as brain capillaries. So these are small blood vessels that are relevant to the control of blood flow. We use spinning discs, so it's old technology that's described in some detail in one of the publications that I have in the references in the beginning. Use the from Andor. And we've been using the same mouse that Elizabeth showed from Mike Kutlikoff with a collaborator, GCaMP2, Connexin 40 GCaMP2, although we can use Flow4 as well. This gives us a resolution of about a quarter of a micron, and we usually image at 50 to 60 images a second. We take the arteries out, we slit them open, and we look at them in en-phos preparation, and we can image about 14 cells in an intact small artery simultaneously. We also freshly isolate endothelial cells from arterioles and capillaries, and are looking at the ion channel currents through in freshly isolated endothelial cells. And in the case of the mesenteric artery in the brain, we can also measure arterial diameter simultaneously because the GCaMP2 is genetically encoded in the endothelium, so we don't have to load with dyes, and we can look at physiological responses such as diameter changes to whatever we're doing. This shows an example of an arterial edge detection. I should also point out that all small blood vessels constrict to intravascular pressure. This is a major regulator of blood flow. It's involved in the brain in autoregulation, so as systemic blood pressure goes up, the cerebral arteries respond by constriction, so that blood flow remains more or less constant. Part of this response is a response to the smooth muscle cells that depolarize and constrict to an elevation in pressure. So in our experiments, we constrict the brain arterioles and other small arteries by intravascular pressure at an appropriate physiological level. This just shows from an article I had in Microscopy Analysis, the spinning disc, and the Andor camera, high sensitivity camera. Spinning disc minimizes laser damage to the tissue, in this case the endothelium, so we can record for longer periods with higher resolution and higher sensitivity. So it's just been really a workhorse for us in looking at endothelial function. A little bit on the GCaMP2 mouse. This shows what we're using now is as Elizabeth mentioned, is a derivative of enhanced green fluorescent protein. It has a calcium binding protein, not calmodulin, incorporated into it. So when it binds calcium, it changes conformation and fluoresces. This has worked quite well, and I'll talk about some of the, or we can talk in the discussion too about some of the advantages. But this all not only genetically encodes it so the biosensor is only in the endothelium, but it also enables really rather long-term stable recordings and homogeneous distribution of the dye. Now, I wanted to tune you into some of the a new grant that Mike Kutlikoff has just received, an R24, and it's involved in the generation of optogenic sensors and effectors in the cardiovascular system. And this shows some of the ones that are some of the transgenic lines that are underway right now, including an endothelial one, FLT1, which is a VEGF receptor that will drive expression of calcium biosensors, red ones and green ones, in all endothelium. And other probes for turning on the endothelium as well as other cardiovascular cell types. I should, I'll mention some other ones. Now to some of the experiments. This will all, hopefully, you'll get a different view of imaging. This is a small artery slit open, and this shows a recording here of the endothelium in an intact arterial wall loaded with flow 4. So you can see some calcium waves and some things going on. On the right is the GCaMP2, and so this shows, I'm outlining here, one endothelial cell. We, on the average, have 14 cells in the field of view. This is the nucleus. And these black holes are positions of a loss of autofluorescence or lack of autofluorescence from the internal elastic lamina. And these holes, black holes, correspond to positions where the endothelium has sent projections through the internal elastic lamina of the smooth muscle. So if we stain these or do them simultaneously, you'll see staining for, and you can see the proteins going through the internal elastic lamina and loss of the internal elastic lamina in these little spots here. So there are 4 or 5 per cell. There are 14 cells in view. And one of the interesting things we noticed right off the bat is that there's little blasts of calcium going off around the projections, these myoendothelial projections. In this case, it turns out to be largely driven by stationary events released through inositol trisphosphate receptors, IP3 receptors, in the projection, around the projection. So this is adding acetylcholine, the classic vasodilator, and as you can see, the frequency of these events goes up. So one of the references, we had a paper in PNAS about, it's now a while back, 2008, characterizing these IP3-mediated events. And we, of course, everybody has to name things. We name them pulsars because they're near black holes, and they happen the same frequency as a celestial pulsar. But one of the things we noticed along the way was that if we silence the IP3-mediated events, this again shows a field of view from a GCaMP2 mouse, and we just added a circuit blocker, we thought this was optically quiescent. We'd gotten rid of all the IP3-mediated calcium signals. And if you look at this image, there were a couple of events, things are running okay, there was a couple rare events. So we then wanted to see, well, maybe these are calcium influx events. And so we had a rather selective agonist for TRPV4 channels. There's transient receptor potential 4 channels. And what you can see is these little blasts of calcium here, here, here. I should put it in this context. This level of the TRPV4 agonist causes a complete dilation of these arteries. And what's driving it are these local calcium signals. And we can also, just skipping, this is shows a arterial from within a mouse brain, isolated, cannulated, and then constricted with pressure, 40 millimeters of mercury. It's 20 microns in diameter, so one of our favorite preps. And what you can see here, again, little holes, and you'll be able to see calcium signals happening in these arterioles too. So this is the sort of thing we're doing now, investigating the Elizabeth's favorite prep, the vascular endothelium in the brain. We also extend this preparation where we can see terminal arterioles come up and capillaries, so we can look at the capillaries attached to this and pressurize it and look at endothelial calcium signaling simultaneously. So what do the signals look like? These are the IP3-mediated events here that I've described. And these new signals that we just found a couple years ago, we put regions of interest, 1.7 square microns, and we look at the change in fluorescence in these regions of interest. And what we can readily see here, the blue ones are the ones induced by the TRPV4 agonist. And what you can see that they're square-like. And what we immediately thought that these events might represent calcium influx through single TRPV4 channels. Now this seems sort of incredulous because it's the concentration of the TRPV4 agonist that causes maximum vasodilation, yet we're only seeing, as you saw in the movie, only a few signals per cell. And then on the average, we've calculated in that less than one channel per cell is involved in vasodilation. And then we named these Sparklets, sort of a historical calcium signaling term, but that corresponded to calcium signal through a single voltage-dependent calcium channel, now the TRPV4 channel. And what you can see here is this shows a snapshot over 94 seconds in the field of 14 cells. And you can see there are about 28 of these sites going off in 94 seconds. And the events are stationary. And if you take the whole cell membranes, these correspond to, on the average, less than one event per cell active. And so what we wanted to do is provide evidence, and so I'll go through some of the criteria, and this has been pioneered by Ian Parker, but provide evidence that we're looking at calcium influx through single calcium permeable ion channels, in this case a TRPV4 channel. And there were a number of criteria. One, obviously, the events have to be quantal, which I'll show you in a second. In other words, each level has to be the same. So this would, in order for the calcium influx to be proportional to fluorescence. The imaging volume has to be very small, in the order of a couple hundred femtoliters, which is what we see in these endothelial cells. And that the activity of the Sparklets, these unitary events, should be affected by drugs that block and turn the channels on, but the quantal level, when the channel opens, it lets calcium through at a constant rate, and that should be unaffected. And so this shows some examples here. The criteria, you have to have a very small recording volume. In the case, the previous experiments that have looked at these events have been mostly done in cultured cells, and they use TERP microscopy and EGTA to sop up the calcium as it leaves the pore. But it turns out endothelial cells are sort of almost turf-like themselves. They're very thin, so they're maximum about 5.5 microns thick. We estimate our recording volume is in the right range where you have enough fluorophore to have a signal, but not too much so that you have a distortion. We have an ion channel, a TRPV4 channel, that has high calcium permeability and high signal channel conductance. And so the prediction would be the quantal levels shown here should, if they reflect calcium influx through an open channel, the levels should go up and down with the calcium electrochemical gradient, which is shown here. And we also depolarize it, it should go down with the electrical gradient, which was not shown, but we did that as well. So the amplitudes are affected by the calcium electrochemical gradient, but this shows an example of the quantal levels. I should say, sort of like Elizabeth, we developed all the sort of analysis programs in order to be able to process these data, these unitary events. And most of those are contained in supplements and two of the science signaling paper I have in the references. But we can see that the levels are quantal, we never see more than 4 levels at one time, and that the adding more of an agonist increases the activity but doesn't affect the quantal level. So it's just like you're turning on an ion channel, but when the channel's open, you have more of them open, but the flux through a single channel remains the same. And then we add antagonists, a couple good antagonists, and the probability of the events, the activity goes down, but the quantal level is unchanged. And what we made a remarkable observation, now we can do basically is this optical patch clamp. So we can image single ion channels in 14 cells simultaneously with high spatial and temporal resolution. This would be equivalent to, if you take each cell has a surface area of 1,700 square microns times 14, this will be equivalent to years of successful single-channel measurements with the patch clamp technique done in one afternoon. And what we saw immediately is, because we can see where the activity is, that they're always in groups of 4, and that the activity is not independent of one another. So the probability of seeing 4 levels is much higher than we expect for 4 independent channels. So we went through this and provided evidence that these things looks like cooperative channel gating that pumps up the calcium flux in these areas. We also used other approaches like coupled Markov chain models that determine coupling coefficients amongst the 4 channels. And the remarkable feature was we found that the coupling coefficients, the cooperativity is the strongest at the projections and weaker elsewhere in the cell. And this translates to an increased calcium influx to projections, which communicate to the smooth muscle. So I'm just, this is showing what, when you have the spatial resolution to see what's going on, you get a lot more information. Just for anybody that's interested, we've been able to look at not only TRPV4 sparklets, V3, which is the one involved in warmth sensing. This is carver crawl. This is oil of oregano. The TRPV1, which exists in some types of smooth muscle in the midmeningeal artery, activated by capsaicin. And we can also break down the IP3-mediated events and see unitary IP3-mediated calcium release events, much like Ian Parker. Then we can then calibrate this to the calcium influx in the sparklet through a voltage-dependent calcium channel, which is purely calcium influx. All these other ion channels let other ions through, so we can look at the ratio of calcium permeability of the different channels in a way that can never be done otherwise in physiological solutions. This is basically an advertisement that this approach can be used for a variety of ion channels that have calcium permeation rates greater or equal to the voltage-dependent calcium channel. So I wanted to show you this example of also what could go wrong in the brain or in endothelium when you have too many TRPV4 channels open. This is also on YouTube if you want to experience it in pseudo-color. And this shows the TRPV4 agonist coming on, causing first sparklets, then calcium waves, then endothelial death. So pretty cool. So this shows you the summary of the data, which we think also applies to the arterioles in the brain. The TRPV4 are in these projections, which and they're activated by endothelial-dependent vasodilators through an anchoring protein that then turns on the TRPV4 channel, lets calcium in, activates calcium-sensitive potassium channels. Hyperpolarization spreads this way. But equally important are the ones that are not in the projections that may be involved in sending signals along the endothelium, as Elizabeth has described. So they're calcium-sensitive potassium channels as well as inward rectifiers in the vascular endothelium. So just to sort of show you where we're moving forward, we now have a new mouse that has two of them, both GCaMP5 M-Cherry biosensors. They're chimeras, and so the red doesn't change fluorescence with calcium. Green does, GCaMP5. So we can now do ratiometric measurements. This is an arterial from within the brain. It's now in the smooth muscle cells. We have the same mouth in the endothelium. GCaMP5 is slightly brighter than GCaMP2. And this shows a 20-micron diameter artery, which is constricted 4AP. You can see calcium going up. And you can now follow the expression of alpha-actin within the penetrating and parenchymal arterioles. And in fact, we can, as you see in the next slide, we can also look, this is from the retina, Alpha-actin is also expressed in some of the contractile pericytes that are proximal to terminal arterioles, and this shows an example of pericyte calcium signaling from the retina using this Acta2 GCaMP5 M cherry mouse. This is just some pseudocolor. So we're now in the position with our brain preparation to look at endothelial calcium signaling capillaries, smooth muscle, make the electrical measurements, be able to track the smooth muscle to the terminal arteriole with the Acta2 GCaMP5 and into the contractile pericytes. And so I didn't talk as long as I don't have to talk as fast as Elizabeth. I had more of an overview. Some take-home points. Local calcium signals to TRPV4 channels control endothelial function. I just gave examples of the mesenteric, the same thing we've seen in the brain with some interesting variations. I should say that we've also patch clamped the endothelial cells from arterioles from the brain, and the TRPV4 current density is the same in the brain as it is in the mesenteric arteries. The same types of sparklets in the brain, same 8-cap150, the 4-channel metastructure is the same. We've also published papers looking at the calcium-sensitive potassium channels, which will be key to translate the calcium signal that Elizabeth talked about into an electrical signal by activating calcium-activated small conductive, intermediate conductive potassium channels. I just described sort of optical patch clamping, which we're now applying to, as you saw from one of the images, to brain endothelium, local calcium signals. There's single ion channels to be imaged in the attacked arteries. You can do them in pressurized arteries as well, but then the planar nature of the on-phos preparation is lost, so you're basically limited to sort of a plane of 2 or 3 cells. You do it in high spatial resolution and temporal resolution. Calcium influx, Sparklets can be measured in ion channels with calcium influx rates greater to or equal than to the voltage-dependent calcium channel. This basically includes all the TRPV channels, TRPA1, which is the irritant, mustard oil, garlic, wasabi sensor. I think the genetically encoded calcium biosensors really enabled us to do tissue-specific calcium imaging in microdomains, and has a number of advantages, including, at least in our hands, homogeneous expression of GCaMP2 and now GCaMP5, little or no toxicity, unlike the AM dyes. Recording times are longer. You lose something in signal to noise, but you make it up for it in many other ways. And if you look at the new R24, you'll see more types of genetically coded calcium biosensors coming online. And in this case, spinning disk microscopy, old technology, but used with new cameras, has enabled longer recordings with higher resolution than the conventional laser scanning confocal microscopes. All this work was just little acknowledgments supported by the Leduc Foundation, the Heart Lung Blood Institute, NIH, American Heart. Swapnil Sankasari has done a lot of the work. Adrian, much like Elizabeth, wrote all their software for the Sparklet analysis, as well as other unitary calcium signals, the bike path in Burlington. The view out my lab window just right now looks the same, frozen. It's about 0 degrees out. This is Mount Mansfield. Work was also some of the other collaborators. Mike Kutlikoff, longtime collaborator. Check out his website. And Vermont. Thank you very much.
Okay, thank you very much, Dr. Nelson, for your presentation. So we have a bunch of questions, and we have very limited time, but any of the extra questions that we haven't got to, we'll send to the speakers, and they can maybe email the people writing the questions. So just to begin with the first question, pretty simple question, could be for both of you. So what's the role of nitric oxide in neurovascular coupling?
Do you want me to answer?
It's a kind of general question. This is either Elizabeth or yourself.
Well, it's pretty. If you know, there are studies where you add NOS inhibitors and there's a decrease in functional hyperemia and neurovascular coupling. It gets complicated quite fast because as eNOS in the endothelium produces nitric oxide, there's nNOS from the neurons. So the source of nitric oxide is not entirely clear. What is clear, I should also say, is that if you isolate an arteriole from parenchymal arteriole from the brain or from the surface of pedal circulation, they will constrict to NOS inhibition. So there's enough NO presumably being produced to cause a tonic dilation. But I think it's really still, I'm with Elizabeth. I think the endothelium plays a major role. So anything that's changing NOS activity could have an effect. But the electrical signaling would have to be through calcium-sensitive potassium channels and inward rectifier potassium channels that are activated during the process.
Okay. All right. The next question is for Dr. Hillman. It is so it's known that astrocytes progressively mature postnatally, and also that the neurovascular response to neural activity is functional until a few weeks after birth. According to Dr. Hillman's and others' work, I would like to know if there is a direct link between astrocytes maturation and neurovascular coupling onset, and whether astrocytes are only able to be to release vasoactive mediators a few weeks after birth. More precisely, do you know when astrocytes start to secrete EETs in response to glutamate? Thank you very much.
Right. Okay. So yes, we have done a lot of work in the last few years, and more to come, on looking at the postnatal development of neurovascular coupling, finding that in the newborn mouse brain, there's really no coupling, that there's no blood flow increase when even when there's a neuronal response, the blood vessels don't seem to know about it. And we're very interested in that process of development and how it comes about and how it establishes, because we think it's actually a really critical component of normal brain development that people don't tend to think of when they're looking at either developmental disorders or how to treat premature infants, for example. And also it's very relevant for functional MRI studies of the developing brain. Now, in terms of astrocytes, I think the general conclusion to draw about what do we know about neurovascular coupling is that we still really don't have the full picture. I would like to think that endothelial cells are a major component. I think astrocytes play some role. It's very hard to understand fully what role they play, whether it's, you have to remember that there's the neurovascular coupling in response to functional activation, but there are plenty of other reasons why the brain might want to change its blood vessel diameter, and I think astrocytes fit into that milieu. Now, we know that astrocytes do develop postnatally. We ourselves have done some studies. We've looked at the astrocyte morphology as a function of development using 2-photon microscopy, and we were quite surprised that in animals that didn't have a hemodynamic response, the astrocytes at least looked like they were all hooked up and connected the way we would have expected. So it would have been wonderful to find that they weren't, and that explained everything. We didn't find that. But there is a lot more to do at this point to really understand, which cells and what that process is that establishes the neurovascular coupling, and that probably will be the topic of one of my future grants.
Okay. For the next question, question came up, is there an emerging big picture of how these signaling pathways are involved in homeostasis and maintenance of vascular health?
I'd like to think that there is a big picture, that they are all tied together. And I think our finding that the endothelium is actually involved really helps to tie those things together, because there's a lot of sort of observations of, somebody who has cardiovascular disease is also experiencing cognitive decline, or somebody who had a bypass surgery or is in heart failure. I think trying to find these common links is really important, because we actually have a lot of treatments for vascular disorders, but we don't use them with any mind of them having any effect on the brain. So my hypothesis at the moment is that this is a sort of unifying idea, but I think there's a lot more work to be done.
Interesting. Okay. Okay, next question. Together with the changes in blood flow, are there also changes in vascular permeability in response to neuron activity?
It's been proposed by the MRI people. There's an idea that there might be water exchange that could sort of account for the required changes in volume of the vascular component versus the brain component. And it's certainly something we've considered as a way in which the brain could change its hematocrit and the availability of oxygen. But to my knowledge, it's not been observed. Mark may know more at the sort of molecular level.
It's possible. Calcium in the endothelium both regulates permeability and the activation of potassium channels and eNOS. But to show that in the brain, in real endothelial cells or blood vessels that permeability is changing at the same time is a pretty. I don't think it's really been nailed down.
Okay.
But I wouldn't be surprised if there's some sort of dynamic interplay. And I would say I also agree with Elizabeth that I think the endothelium is sort of like the shadow nervous system of the brain. Mm-hmm. It's like these are basically wires through the brain, and they're sending signals, transmitting signals from the brain interior to the surface. And the question is, how is that happening? And really, more of the mystery too is what is happening in the capillary endothelium. Mm-hmm. How are they electrically coupled? What is generating the electrical signal if it's coming from there? So there are a lot of really basic fundamental questions that are about the circuitry and how this is working. And of course, as Elizabeth showed in one of the cartoons, that all the things that are in the influx seem to be in the endothelium. And in the case of the arterioles, you got a little sandwich. It's sandwiching a little of the smooth muscle bologna there, and so anything that would activate is released. Anything that changes within the brain, if there are relevant receptors and/or ion channels in the abluminal surface of the endothelium, it's going to be sensing it, and I think it's almost incomprehensible to me that it's not somehow an active role in functional hyperemia and controlling blood flow, although it's been viewed mostly as, until Elizabeth, as a passive process.
Okay. So I think so there's a bunch of other really good questions. Unfortunately, I've been told that I have to wrap it up right now. So once again, I'd definitely all the questions. We'll forward these to the speakers and get some answers to those. And so then anyway, I'd like to take this opportunity to thank all the speakers, both of the speakers for their presentations, and also thank the attendees for today's webinar. This has been really interesting. I learned a lot, and thanks for your questions and also your answers. So also, don't forget there will be a recording of this webinar that will also be available online very shortly. So thank you very much, and have a good day.